That anybody who works with cells, whether they are primary cells or cancer cells or cell lines, is to split cells or passage them or to place them in different plates or dishes. One thing that you need to make sure of is that your biosafety cabinet has the UV on for at least 15 minutes before you start. As you can see, the UV has been on for at least 15 minutes. So we'll start passaging cells or splitting cells. The first thing we do is to spray and clean the biosafety cabinet hood with 70% ethanol. Anything that goes inside the biosafety cabinet has to be wiped with ethanol. So I have phosphate buffered saline that is sterile. I have the growth media, and each cell line, each primary cell type has its own growth media, and I have trypsin, an enzyme that we use for splitting cells. I usually split cells when they are 70 to 80% confluent. So let's take a look at these cells. These are breast cancer cells. You can see they are epithelial cells. And I can, this is the 10x, let me use the 4x so that you can see the confluency. So you can see that the cells are 60 to 70% confluent, which means the cells are occupying 60 to 70% of the field. And this is very good for you to split cells. More than 90% you will start having altered gene expression, altered metabolism, and we don't prefer that because you are changing the identity of the cells you are working with. Okay, so we'll start passaging or splitting cells. So what we will do is aspirate the media and wash the dishes twice with BBS, phosphate buffered saline, the sterile buffer. And the idea to wash is to get rid of the serum because cells have been grown in serum. So we'll aspirate. And again, everything is sterile. Make sure that the tip of the Pasteur pipette does not touch anything. It's sterile. It has been autoclaved. I will wash twice with 6 ml BBS each time to get rid of the serum. The growth media has serum. Serum has all the growth factors needed for cell viability. So this is one time. This is the second time. And now I will add trypsin. And trypsin is an enzyme that breaks down proteins. And these proteins are the ones that keeps the cells attached or adhered to this plate. So trypsin is a protease, and we buy it as 10x. So I'm diluting it, 1.8 ml BBS and 200 microliter trypsin. So I added 200 microliter into the dish that has 1.8 mils BBS. So the total is 2 mils or a 1x dilution. We'll leave it in the incubator at 37 degrees for 3 to 5 minutes. until the cells start floating. More than 5 minutes, this will cause toxicity for the cells, and the cells will die because, again, trypsin is a general protease that will start not only breaking down the focal adhesion, but it will start working on the cells themselves. Meanwhile, you can prepare your plates. So these are tissue culture plates. Everything is sterile. So you will open it here. And also I want to grow the cells in 24 well plates. I will bring one. This is 24 well plate. It has 24 wells, and we use such a plate if we have different treatments so that we can have duplicates or triplicates of the treatments. One thing I like to do is just to try to dislodge the cells and not be too aggressive. I just knock the plate a little bit. Let's take a look. Okay. Can you see that the cells are floating? Which means they have dislodged. You see? Which is good. So, in order, again, trypsin is toxic. In order to end the trypsin reaction, we will add serum. Serum has anti-trypsin activities. So I will add 8 mL of the growth media that already has serum. I will add 8 mL. That will give me a total of 10 mL. So I have these 24 wells and I have that plate. What I will do is I want to split my cells or passage them into a new dish, because again, if they are over confluent, you will lose the identity of these cells. And I want to split them 1 to 10. So I added 1 ml of media, to which I will add cells for my next experiment. Okay. So I have this plate, the new one. I will add 1 ml of the old plate that has 10 mL of floating cells. This tip cannot go to the regular trash. It has to go to the trash bag that will later be autoclaved because it has cells. In order to finish, you need to write the name of the cells. So mine are MDA MB 453. Passage number, how many times you have done this passaging experiment. So this is passage 21. You need to write your initials and the date. And now you can put them back in the incubator. Putting them back in the incubator, you have to clean the biosafety cabinet. So the pipette, this is I use this pipette with cells, so you cannot throw it in the regular trash. Again, it has to be autoclaved. This plate had the original cells, so we will aspirate what is left of the cells. Then you will bleach the dish with 20% bleach. And again, the plate has to be autoclaved. Make sure that everything, all the bottles you have used are tightly closed so that they will remain sterile for next use. So I made sure that they are tightly closed. And now for the aspiration line, it is already bleached because of the plate. Now you will aspirate some 70% ethanol into the aspiration line. The Pasteur, the sterile Pasteur pipette has to be autoclaved. So we will autoclave it later. Turn off the vacuum. And now you need to ethanol, clean everything just like when you started. And also you need to clean the glass sash with 70% ethanol. Turn on the UV for 15 minutes. And that would be the end of cell passaging.